So during the end of World War I, there was a pandemic flu circulating the globe, actually killing millions of people. It was called La Gripe, or Spanish flu, mainly because it killed a lot of people in Spain early on during the infection. And what you can see here on the left is just basically a medical tent. So this virus hit the United States pretty hard, actually, first in Kansas, and then actually jumped over to Boston about a year later and killed a bunch of people here. And I've taken a little excerpt from the Journal of American Medical Association, which pretty much says, we've done a really good job killing each other during the war, but we should really focus our attention on Mother Nature, which is doing a much better job at killing us. And to highlight this, during the war, it was about 8 to 13 million estimated combat deaths, but the Spanish flu is estimated to kill between 20 and 40 million people in about a year or two. So it just highlights that this virus is able to spread very rapidly and kill lots of people, and no one was really studying infectious diseases, especially viruses, at this time. And so what's really interesting is that people didn't actually isolate the fact that this disease was caused by a virus till 1933. We sort of just got lucky. The pandemic just sort of died out around 1919, stopped killing us. But it really highlighted the fact that we should probably study these viruses, because obviously we don't want millions and millions of people to die without us knowing what's going on. And sort of segues into research was actually able to track down avian influenza, which is just like human influenza but infects birds, to these two lakes in China called Qinghai Lake and Poyang Lake. And in these lakes you actually have thousands of migrating birds that pass virus to one another and then actually begin to spread it worldwide. So what I'll show you here is you have these sort of two nucleus of avian bird flu. And these migrating birds migrate all over the world, and as they migrate, they actually spread this avian flu to other birds. And as you can see, there's sort of a cyclical cycle, which is why it's the flu season. And I want to really stress that avian flu is not human flu. So we get a different type of the flu. It's the same species of virus, but it has to actually jump from birds into humans. And the CDC actually uses this data to monitor human flu trafficking. And this is actually how the flu vaccines that we get every year are created, because they actually see, oh, this flu's coming over here, maybe we should vaccinate. However, you can see that there was 2,000 different types of influenza viruses in the United States just last year. So it's pretty hard to guess. Your flu shot only has about 3 or 4 different flu viruses in it. But so my main part that I'm going to focus on is telling you guys what a virus is and how diverse these are. There's tons and tons of different viruses. There's not just one type of virus. And then Joe's going to come in and tell you sort of that second part, where a virus can actually jump from an animal into a human, and that's what's called zoonosis. So what is a virus? If you guys leave this talk with anything, I hope it's this, that a virus is just a little packet of protein that encapsulates a virus genome. And a virus's genome can be RNA or DNA, like we just learned about in the central dogma. And so to put it in context of how big a virus is, let's take a flea, not that flea. And a flea is about 2 millimeters in length. And if we were to zoom in on that flea to see one of that flea's cells, we'd have to zoom in about 100 times. So a normal cell is about 20 micrometers. And a micrometer is just 1/1000th of a millimeter. And a virus is even smaller than that cell. So we'd have to zoom in another 100 times. So most viruses are in this range called 200 nanometers. So a nanometer is 1/1000th of a micrometer, 1/1000th of a millimeter. And so what that tells you is that a virus is about 10,000 times smaller than a flea. And to put that in sort of a human scale, if you were driving along in a sedan, like a Camry, a quarter would be about the size of a virus. So these very small agents, which is just protein with some RNA or DNA inside, can actually cause these horrible effects to us. And I also want to stress that not all viruses look the same. So these are some of the smaller viruses. So influenza A, which I'll talk about later, causes the flu. Hepatitis C, which can cause liver cancer and hepatitis. And this little phage here, which is a lot of people like to use. It looks like a lunar lander. And these are interesting that they only infect bacteria. And I'll get into that in a second. Next up on the sort of size spectrum is everyone's favorite herpes. And there are actually some very large viruses which were recently discovered, and they're named mimivirus because they mimic a bacteria because they're so large. It's not because they're small. And there's also one called megavirus. And these are also phages that only infect, actually they infect fungus, not bacteria. But anyway, so what's the daily life of a virus? So the main thing a virus wants to do, it's a very simple idea, is just get in a cell and make more virus. There's nothing else a virus wants to do than just make more virus. But to get in a cell is actually sort of tricky. So cells have specific receptor proteins. So earlier on we were talking about the low-density lipid receptor for cholesterol, and that was a receptor that had to bind to this low-density protein. So think of a virus in the same sort of way. So it's surrounded by proteins, and these proteins have to interact with a specific cell protein in order to get into the cell. Once a virus is in the cell, its next step is pretty easy. It just wants to make lots more virus. So it does this by making virus proteins and its own genome. And once it's done this, it encapsulates those genomes with the virus protein and leaves the cell. And it's actually when the virus leaves the cell that most of your cells will actually die. And so going back to that sort of first part of how does a virus get into a cell is this concept of tropism. So it's a nice word you can use from now on when you go and you have your little dinner parties after this. You can talk about tropism. So tropism in its simplest sense is that a cell has to, in order for a virus to get into a cell, that cell has to have a specific protein that a virus can bind to and get into the cell. And so we have lots of different cells in our body. So what that really means is that not all viruses can infect all of our cells, and more importantly in human biology, just because one virus can get into one of our cells and infect it doesn't mean it can infect all types of our cells. So let's take the liver again. We have liver cells, but you also have cells in your eye. And these cells all have the same DNA, but they don't all express the same proteins, because obviously you don't want liver function going on in your eye, and you really don't want to see what's going on in your liver, especially after you drink. So you can think about it like this. So hepatitis, which is a virus which only infects liver cells, can't actually infect your eye, even though the cells are essentially the same because they have the same DNA. But they express different proteins which allow the virus to only infect a certain one. And that's the idea of tropism. So to illustrate that a little better, we've brought back the bacteriophage. So bacteriophages can only infect bacteria. And that's because bacteria express certain proteins on them which allow the phage to get in. So it can make more virus. However, the human cells don't express the same bacterial protein. So even if you got a bacteriophage into a human cell, it actually wouldn't be able to make more virus. And likewise, we have our favorite virus to contract at Disneyland, measles. And we all know measles can get into human cells, but bacteria don't express the same proteins that allow the virus to get into them as human cells, so you can't get into them. And what's really interesting that Joe's actually going to touch on is that this tropism is all still based on the virus. So the virus can mutate its proteins to actually jump hosts. And that's what a zoonosis is when like you have a bird flu, which usually only infects birds, able to jump into humans. And that's a zoonotic shift. And that's also really related to tropism. So we all have lots of different cells, and I'm just going to be frank, there's millions of different viruses that infect all your cells. So if there's millions of different viruses and lots of different cells, viruses have to be very diverse. And they're sort of broken into 7 orders, which are just large groups. And then they're further differentiated into families. And don't bother reading all these. They'll be up on the slide if you want. But the Filoviridae family is what Ebola lies in. So now you can tell people, oh, the Filoviridae outbreak in Africa was really bad this year. Picornaviridae is actually the smallest of all the viruses. That's why it's named Pico. And that's actually where polio comes from. So the sizes don't really matter on how bad the virus is. And then, interestingly, within each of these families, you have species, so several species of virus. And each species, you can have thousands of strains. So like flu virus, that was all influenza A, what I showed you earlier, had 2,000 different flus just in the United States. Those are all different strains of influenza A. So they're all the same species of virus, but there's thousands of different strains. And a strain is just sort of defined by like a single mutation within the virus. And so I'm going to break it down a little simpler for you guys because I don't want to talk about orders and you don't want to remember strange Latin names. So the central dogma that we learned earlier shows that you have DNA, which encodes for RNA, which encodes for a protein. And you can sort of take the virus take on this. So you have viruses with DNA, which are called DNA viruses, like herpes simplex. So the herpes simplex is a DNA genome that encodes for its viral RNAs that encode for its viral proteins. However, you also have viruses that are just made of RNA. So influenza doesn't have any DNA at all. It just comes with RNA to make its virus proteins. And in contrast to the central dogma, which was actually really controversial when it was discovered, you have the retroviruses, which actually are made of RNA, and they have a protein called reverse transcriptase that actually makes RNA into DNA. So it was actually pretty controversial when they found this because everyone was like, oh, the central dogma, that's the only thing that can happen, DNA to RNA to protein. And then virologists came along and like, no, you can have RNA, it can go into DNA. And then, so virologists like to break the rules. So in summary of this first little part, I just want to re-emphasize that a virus is just a little packet of protein with some genomic information in it, and that it has to get into a cell to make a new virus. And in order to get into a cell, there's a concept called tropism, which is a virus needs to infect a cell that expresses specific proteins which allow that virus to get into it. And the last real point is that viruses are really diverse. So I'll take any questions, and then I'm going to move into 3 examples of DNA viruses, RNA viruses, and viruses with reverse transcriptase.
So we have bacteria that are required.
Yes, we do.
Our bodies can work.
We have more bacteria in our bodies than humans.
Do we have
cells.
viruses in our bodies that our bodies required to work?
So Joe actually studies this, but we've evolved over time to actually incorporate virus genes into our cells. So like the cancer talk didn't discuss this gene called MYC, but there's actually a viral MYC, which is one of the first oncogenes discovered. So a lot of these early genes that were discovered for cancer were actually found in viruses. So we know that we have viruses that give really bad things to us. Viruses that give really good things to us are still studied a lot, mainly because it's hard to tease out which came first, us or the virus. But I know that herpes can, if you're infected with herpes, you have less chance of dying from leishmania, if that helps. But it's not a direct, we don't really know if we need viruses in order to survive. All right, so I know I rushed through that because I'm nervous, but I'll try and slow down for this next one. So viruses in focus. I wanted to focus on herpes, which is a DNA virus. And DNA viruses are usually much larger because DNA as a molecule itself is actually very stable. And so going back to the central dogma, we know in order for DNA to have any function, it makes RNA, which then makes protein. And since viruses that have a DNA genome, it's a double-stranded molecule, it's very stable, they usually don't mutate as frequently. In contrast, we have RNA viruses, which their entire genome is RNA, and when you make more RNA, you're more prone to put in errors. So you get lots more mutations. So RNA viruses actually mutate far more frequently than DNA viruses. And a lot of the pathogens that you hear about in the news are actually RNA viruses. So Ebola, flu, and well, polio, but no one gets that unless you're in India. Or not India. India's been polio-free for 2 years now. So. And then the last virus I'm going to tell you about is HIV, which is a retrovirus. I'm really going to focus on the differences between how a retrovirus has to take its RNA, turn it into DNA, and then insert that DNA into the host DNA before we make more viruses. It's much more complex than a lot of these other viruses. But let's focus on herpes, which I study, so it's very close to my heart. And unfortunately, it's on a lot of your lips right now because 60 to 80% of the population in the United States actually has HSV1 in them. And the reason it happens is there's this sort of iceberg effect, where a lot of viruses will infect people, but only a small, like the tip of the iceberg, will actually present symptoms. And so I was talking about tropism earlier. HSV is a neurotropic virus. It likes to sit in your neurons. So it comes in usually through your face or your genitals, and it actually travels through your neurons back into your spinal cord, where it sits, and it can sit for years at a time. And while it's back there, it can either make lots of virus, which will come back through your neurons to present those cold sores you get, or it can just sit there literally until you die. It won't make any more virus. It's sort of happy to just sit there. And although cold sores are very annoying, herpes can actually be quite dangerous and deadly. So there's herpes keratitis, which is an infection of the eye, which if unchecked can actually lead to blindness. And that's because you have an immune reaction that actually goes in and starts killing your eye cells. And likewise, you have herpes encephalitis, where if the virus goes into your neurons and instead of just lying low, starts making lots of virus, it can actually promote the immune response to start attacking the tissues in your head, which can cause inflammation of the meninges, which is a tissue around your brain. So delving a little further into herpes virus, I'll give you a nice example of tropism. So the herpes virus uses this receptor called nectin-1. It's just a protein on your cells, and unfortunately it's on a lot of your cells, especially your neurons, which is why it's a neurotropic virus. And so in order to get into the cell, the herpes virus protein binds to this nectin-1 and then goes into the cell's nucleus. And once the virus gets into the nucleus, it starts making more virus. And the way it first does that is by making these what are called immediate early proteins. And these proteins go around the cell and sort of prep it. So you can think of a virus as sort of infiltrating a cell and turning that cell, which would normally just do normal cellular activities, into a virus factory. So these immediate early proteins go and they shut off your immune system, and they basically tell all the cell's proteins, hey, look, it's time to make virus. At which point you get these early proteins, and all the early proteins do in a herpes virus is go into the nucleus and make lots more DNA, lots and lots of DNA. So this entire cell is getting converted basically into just one large house to make viruses. And at the end, once you've made enough new virus DNA, it's time to get out there and infect someone else and give you a nice cold sore. And to do this, you have what are called these late proteins. And the late proteins are what make up the capsid, or the actual virus. And they go around and they encapsulate the virus DNA, and then they leave. And so what can actually make herpes virus dangerous? So I told you earlier about these early genes, which are just involved in making new virus DNA. And so one of these, if these genes are too active and you're getting way too much viral replication, you'll spread to a bunch of other cells, and obviously you'll start killing cells that you don't want to die, like your neurons. And so one of these genes, herpes thymidine kinase, basically just sees DNA and has to modify it in order for it to be incorporated into the new virus DNA. And researchers have actually been able to find this drug called acyclovir, which looks just like a DNA particle, but it's missing a little piece that basically doesn't allow you to incorporate more DNA. So once you add this into a herpes virus DNA, the virus is essentially dead. It can't make more DNA. And so thymidine kinase is a very greedy enzyme. Like most proteins in viruses, they're very greedy and they just want to make more virus. So this drug actually tricks the virus into incorporating this poison DNA into its own DNA and stops the virus from growing. However, earlier we were mentioning mutations causing resistance to cancer drugs. Viruses are in a constant battle with the host immune system and the cells that it infects, as well as drugs we give it, to evolve mechanisms to overcome this. So there's actually a single mutation that can occur in this thymidine kinase gene, which stops the virus from being able to incorporate this poison. And it basically leads to a herpes virus that's resistant to the drug. And this is really dangerous if you're having an encephalitic outbreak, because now we don't really have a drug to treat the infection. So speaking of mutations, let's move over to RNA viruses. So influenza, everyone's favorite virus to take a day off work. And RNA viruses mutate a lot more than DNA viruses. And this is essentially because they have an RNA genome. And RNA is much more easy to mutate than DNA when it's being made. And so a little interesting factoid, when we hear about flu, we usually hear H1N1, H3N5, and those are actually referring to the proteins on the surface of the virus, and that's how they're named. So the hemagglutinin protein is involved in the virus's tropism. So in order for this virus to get into a cell, its hemagglutinin protein has to bind to a cell receptor in order to get in. But the N comes from this neuraminidase, which is actually involved in cutting the virus loose. So when the virus actually leaves a cell, it has to get cut loose because it just is sticking to the surface of the cell. And so like an H1N1 virus will have a hemagglutinin type 1 and a neuraminidase type 1 protein. Now you know. And what's really interesting about the flu is, unlike herpes that only had one genome, just one piece of DNA, flu actually has 8 different genomes. They're all RNA, but what makes flu able to mutate so rapidly is that these genomes can actually be mixed and matched. So if you had 2 viruses infect the same cell, say one of them was H1N1 and one of them was H3N3, you could actually get a number of viruses that are now H1N3 or H3N1, etc., etc. So it actually leads to a lot of new flu viruses being developed, or being created. So the average life cycle of avian influenza is in migratory waterfowl like ducks and cranes. And so they go and they sit in these lakes, and the flu virus is actually very stable in a lot of these alkali lakes. And they pass virus from one another. They can then pass it to chickens and so on and so on. And while they're inside their hosts, they have these 3 proteins that are made, each one from its own separate RNA. And these are called PB2, PB1, and PA. And these are the proteins that actually help the flu make more RNA. So they make the genome of the flu. And interestingly, PB2 is able, if it mutates, just a single little tiny bit of the protein changes, it can actually now jump from birds into humans. And so this what's really fascinating is that this is the same mutation. It's called PB2E672K if you're really interested. But this same exact mutation was found in the pandemic flu of 1918 and 19. And so it's also been linked to a more deadly flu. And through actually researching what this mutation does, scientists have been able to find out that it actually causes the protein to make lots more RNA. So it makes tons more virus. So in the last couple minutes before I hand it off to Joe, I just want to go over HIV, because it's a very important virus. It affects a lot of people around the world. And actually, interestingly, most research into viruses is actually focused on HIV today. So the main thing you should know about HIV is it has an RNA genome like the flu. It's only one piece of RNA though, not 8. And it has to actually convert this RNA into DNA before it can make more virus. So here we start off with the HIV RNA. We're already in the cell. And it has to make DNA from its RNA. And it does this with a protein called reverse transcriptase, which goes along the RNA and actually makes DNA that complements it, and then makes a double-stranded DNA. It then actually has to insert this DNA into the host genome. So just because it has this virus DNA doesn't mean it can make virus. In order to make virus, it has to actually get into the host genome. And to do this, it uses a protein called integrase, which basically splits open the genome of your cells and inserts its virus DNA right in there. And once this occurs, you can actually have HIV making new particles, or what HIV tends to do is sort of just hide out in your cells. So HIV infects your T cells, which is why you have this immunodeficiency, is you eventually lose all of your white blood cells. And this is sort of shown here, where in red you have the number of viruses, and in blue you have the number of T cells. And so initially when HIV infects and inserts itself into your T cells, it starts killing a lot of your T cells and making a ton of virus. And these viruses go out and infect lots of other T cells. But you can see they sort of stop making virus. And so you get this gradual tonic release of virus as you slowly make virus and you slowly lose your T cells until you eventually get AIDS. And then you usually die from a bacterial infection. Luckily now we have highly antiviral retroviral treatment called HAART, which can actually prevent the virus from leaving the cell and making new viruses. So I know there's a lot of information, but to summarize it all, I really want to stress that viruses are always in a constant state of flux. They're always evolving to resist your immune system, to resist drugs, and that's true for bacteria, which we'll hear about later, as well as cancer. There's always this constant natural selection to keep on surviving, make more virus. And then just to go over the simple groups you can remember when you're talking about viruses is that you have DNA viruses, which are made of DNA, they have DNA genomes, RNA viruses, which are made of RNA, and the reverse transcriptase, or retroviruses, which have a reverse transcriptase protein that make their RNA into DNA. And I'll take some questions before we go into Joe, who's actually going to focus mostly on the jump that I was discussing between flu from birds into humans. And yeah.
So, 3 different kinds of viruses, right? Is one group or another more difficult to treat?
They're not more difficult to treat, I would say. So DNA viruses tend to encode more proteins. So DNA viruses tend to be able to better mask themselves. They don't essentially, there's not one specific protein you can usually target in a DNA virus. Herpes virus is just one example of one protein that's really easy for us to target. RNA viruses are easier to target because they usually only have about 8 genes in them. They're very small. However, they mutate so rapidly that as soon as we find a drug target, they sort of go, oh, okay, well, now I can just change my protein and mutate it away. But the RNA viruses tend to cause most human disease, like when you get really sick. There's a lot of DNA viruses that cause disease too, but they're usually not the pathogenic, kill lots of people. Although smallpox was a DNA virus. Yeah?
You said poliovirus is an RNA virus, but it's real stable and the vaccine keeps working.
Yeah, so just because, so when I say an RNA genome is unstable, I don't mean that the RNA and the virus is falling apart. It's when a virus gets into a cell and it's making tons of its genome. You get many more mutations in an RNA virus because when you incorporate RNA nucleotides into each other, the polymerase, which is the protein that makes the new RNA, is less it's less efficient at sensing mutations. So that's what I mean when I say less stable.
It's so interesting that we can use the same polio vaccine for generations, but we have to make a new flu shot every year.
Yeah, so that's the whole sort of catch-22 with vaccines is like rabies, polio, smallpox. We were able to make, a couple viruses and they were stable. Same with measles. Measles is actually right now evolving away because people aren't vaccinating. But immunology is a lot of times referred to as black box. So we got really lucky with rabies. It was just basically, I think it's Pasteur injecting rabies into dogs and then into rabbits, and then we just got a virus that was attenuated and he injected it into orphans. And he was like, look, these orphans don't get rabies anymore. Yeah, it was before the IRB, which is what you have to go through now to get approved through all your study. This was before clinical trials. So that's one, you can thank bureaucracy for that. That's, no orphans get injected with these anymore. All right, but I'm gonna wrap it up because I'm way over time, and Joe's gonna teach you all about the interesting jump.
Thanks. This is the other one. Green. Test, test. All right. Yeah, that's great. So, just getting hooked up here.
Just make sure it's off.
This one? Yeah. Great, all right. Thanks, Cole, for the introduction to viruses. And in regard to that question earlier, are there any beneficial viruses? I think that's the question I've heard. Actually, there are examples in placental mammals. It seems that placental mammals have actually adopted certain virus proteins for the generation and maintenance of the placenta, and they actually seem to be absolutely necessary in some animals. But that's not what I'm going to talk about right now. So Cole I'd be glad to answer more questions about that later. So Cole has introduced you all to the basic tenets of virology. And he's talked a lot about the microscopic details. And for this part of the talk, I'm going to talk about viruses on more of a global perspective and talk about really where do the viruses that infect humans come from. So the current Ebola outbreak has infected more individuals than all previous Ebola outbreaks combined. It's still going on now. Liberia has been recently declared Ebola-free, but it's still in Sierra Leone and Guinea. Additionally, there have been 7 times more deaths in this one outbreak than all the other ones combined. And this is dating back to 1976, when we first discovered this. Fortunately, Ebola outbreaks are rare. And one of the reasons this is because Ebola is not actually a human virus. Like many emerging viruses, it hides in a different animal until by chance it comes into contact with humans and can then start an infection. This is what scientists refer to as zoonosis. So zoonosis, or a spillover, think of viruses spilling over into humans from other animals. We usually only hear about viruses when they cause disease in humans, but everything comes from somewhere, and viruses are no exception. Of course, zoonotic events aren't limited to viruses. In fact, the bacteria responsible for the bubonic plague that wiped out 1/3 of Europe's population in medieval times comes from a bacteria that circulates in fleas. The bacteria that cause Lyme disease and the prion that cause mad cow disease are also considered zoonoses. The rabies virus is a prominent zoonotic event that Cole alluded to earlier, and still causes 55,000 deaths each year, mostly in children. And like I said on the last slide, Ebola is also a zoonosis. So scientists estimate that more than 6 out of 10 pathogens that infect humans are zoonotic. So many diseases you have heard of, and some you may not have heard of, come to humans from animals. These include anthrax, chikungunya virus, African sleeping sickness, Middle Eastern respiratory syndrome, Marburg, Machupo, West Nile virus, Lassa fever virus, Nipah virus, dengue virus, Hanton virus, monkeypox, influenza virus, and SARS. This is only to name a few. But I think you get the point that some of the most problematic infectious diseases that we're faced with right now come from animals and are viral in nature. So viruses seem to have a special potential for zoonoses. Let's look at some reasons why. So as Cole was telling you about, viruses not only have the smallest genomes of the pathogens that cause zoonotic disease, but they also have the highest mutation rate. This high mutation rate offers viruses the ability to adapt. For example, during a spillover event, some viruses may acquire mutations that allow them to proliferate in a new host species, such as humans. The probability of this happening in viruses is much greater than it is in bacteria or parasites. Additionally, viruses are unaffected by antibiotics. Antibiotics are great because they can affect a wide number of bacteria and kill them. However, no broad-spectrum antiviral that would kind of have the similar effect as antibiotics on viruses exist. And a lot of this is because viruses are so diverse, which you just learned from Cole. Additionally, viruses represent some of the most deadly pathogens we know of. In fact, rabies is 100% fatal in humans if treatment is not administered before symptoms. So, another major reason that zoonotic events are so difficult is because they hide between outbreaks. These pathogens hide in what is known as a reservoir host. A reservoir host is an animal that chronically carries a pathogen, but is usually not harmed by it. Pigs are the reservoir for the H1N1, commonly referred to as swine flu, most recently famous in 2009. Birds are the natural host for the West Nile virus, which is increasingly becoming more of a problem. Interestingly, bats seem to be the reservoir host for a whole slew of viruses, not limited to just this list here. It's thought that they might even be the reservoir host for Ebola. So, the path to humans is not always direct from the natural reservoir, the reservoir host, to humans. Many times, another animal, known as a vector, is responsible for transmitting the virus to humans. This is the case for the West Nile virus, which is transmitted from birds to mosquitoes and then to humans. Additionally, some zoonotic viruses need what is called an amplifier host. These are animals that get infected by other animals and then pass the infection on to humans at rates that would not be attainable otherwise. Hendra virus, a very deadly virus in Australia, came to humans through horses that were infected by bats. So additionally, besides the characteristics of a virus that make them highly likely candidates for spillovers, there are other factors that influence the spillover potential of a virus. The first, and arguably the most important, is exposure. What this boils down to is that the more interactions humans have with the animals that harbor these pathogens, the more likely it is for spillover events to occur. This is becoming more of a concern in parts of the world where we're sort of infringing on these areas where a lot of the animals that harbor these pathogens exist. In parts of the world where humans rely on meat from wild animals, the events are happening more frequently. So imagine living near a jungle in a poorer part of the world. One day you go out looking for meat so you can feed your family. You come across a dead chimpanzee and it feels like you just won the lottery. Unknowingly to you, this chimpanzee died of the Ebola virus. A day earlier, an infectious virus is still present in its blood. You go home to butcher the animal before cooking it, and you accidentally nick yourself with a knife. That doesn't seem like much of a problem, but now the Ebola virus has a direct route to your bloodstream to start an infection. At the end of the day, it was the virus that won the lottery. And what I really want to stress here is that these sort of events are really sort of lottery events for the virus. This is just one example, but you can imagine other ways in which humans come into contact with animal viruses. So another key factor that affects whether a virus can spill over into humans is if it can start an infection in human cells. So as Cole was saying earlier, he introduced this key idea of tropism. So even in the event that you come into a virus, if it can't infect human cells, it will not lead to one of these spillover events. However, if there is a match, then this is what can start a spillover. Additionally, human-to-human spread is a major factor that can determine the outcome of a spillover. In our globalized society, it is much easier for pathogens to be bystanders on a transcontinental flight and thus reach parts of the world that they would not have been able to reach otherwise. So luckily, not every spillover event results in the outbreaks that we most often hear about. Most of these events are known as dead-end spillovers. This is when a virus spills over into a human, but either kills the human, he recovers, or never even knows he has the virus. However, if the virus is efficiently spread human to human, this is when we have an outbreak. This can be anywhere from a few people to thousands of people. The Ebola virus of this year and last year is considered to be an outbreak. These are usually confined to specific regions, and over time will burn out. Rarely, a zoonosis can go on to become a pandemic, meaning that it is spread efficiently human to human and travels around the world. This was the case most recently for the 2009 H1N1 swine flu. So we've talked about viruses and how they spill over into humans, but what can we do to limit or decrease this from happening? Unlike viruses that are specific to humans, such as the smallpox virus or varicella zoster, which causes chickenpox, or the polio virus, zoonotic viruses are extremely difficult to get rid of completely because they exist in these other hosts. So the smallpox virus was eradicated completely in 1980, and this is a great achievement. But these zoonotic viruses, what we would have to do to eradicate the virus completely is not only would we have to vaccinate a number of humans against them, we'd also have to rid these natural reservoirs of the viruses. Therefore, so it's difficult to do this because you can imagine a virus that hides in bats. You would have to trap all these bats and vaccinate them sufficiently so that the virus cannot be spread. So therefore, it's important to prepare for outbreaks before they happen of these zoonotic viruses, since they're so difficult to eliminate. How exactly are scientists preparing for the next big spillover? Mainly by surveillance methods. Pictured here is a scientist named Nathan Wolff. He is shown convening with a hunter in Central Africa, a region of the world that is a particular hotspot for spillovers. What he does is he provides hunters with little cards, pictured here, and on the cards you see drops of blood. He then asks for drops of blood from these animals, any animals that the hunter collects. From these blood samples, Dr. Wolff and other scientists can test to see if there are any new or known viruses. This can give clues into what the most likely candidates are for a spillover event, and then scientists can study these viruses in the lab to learn more about them and develop tools to fight them. Another preventative method is to find the natural reservoirs. So finding the natural reservoir is important for eradication and prevention efforts. If the reservoir of a virus is known, efforts can be taken to limit interactions with this virus and lessen the chance of spillovers. Since we can't ask animals what, if any, symptoms they have experienced that would lead to the prognosis of a disease or illness, scientists have to look for hallmarks of viral infection in the animals by trapping them and collecting specimens. The gold standard for doing this is to find the actual virus of interest. One way this is done is by taking samples and seeing if there is any infectious material in them. However, this will only work if an animal is infected at the time the specimens are collected. Sometimes infectious virus cannot be found in a reservoir, but there are other clues that allude to an infection. The presence of antibodies to a specific virus indicate that an animal was likely infected by that virus at some point in the past. This is because antibodies serve as molecular memory to a specific pathogen. Once made, they are long-lasting and can be effective at indicating an animal was infected by a virus. Importantly, antibodies indicate that the animal survived the infection and thus is a candidate as a natural reservoir. Spillovers, like the one that caused the most recent Ebola outbreak, are inevitable. However, by identifying what viruses are most likely to spill over, and by understanding from where these viruses come, we can have the foresight needed to combat them and not be caught off guard. This will allow for quick responses that can halt the virus in its tracks. So the key points from this part of the talk are that most emerging diseases come from animals. Viruses, out of these, seem to be the most problematic for the reasons I pointed out earlier. There are many factors that influence whether a virus will spill over into humans, both human and viral factors. And tracking spillovers and finding natural reservoirs is key to prevention and preparedness. So with this slide, I'll take any questions. Yes.
Have you heard about the new test that's supposed to identify like almost every virus a person has been exposed to based on antibodies?
Yes. So the question was, have I heard about the new test that supposedly can identify every virus based on antibodies? Yeah, so that's a cool technology, and that seems so kind of in terms of what I've been talking about, that wouldn't identify any new viruses, of course, because the antibodies recognize a specific part or a specific piece of the virus. And so for this test, we can only use pieces of viruses that we already know about. So any other ones are yet to be discovered and could then be added to the test. But at this point, it kind of wouldn't work.
When you discussed Ebola, you said that the current outbreak had more cases and fatalities than all the others put together.
Yes.
Is that something to do with the virus specifically, or is there something to do with, are we better at sensing Ebola or detecting it now?
I think a lot of it has to do with the location this time. So most previous outbreaks have been in isolated communities or isolated villages and more remote regions of Africa. So this one was transferred to large cities, and then from there, there's obviously more human-to-human contact, more potential hosts. So I think that was one of the major factors in that.
Great, okay, so we're gonna wrap up, but Joe